In this video, we will discuss about stamen, also called as a microsporophyll. We'll discuss in detail what is the structure of stamen, how it adheres open, breaks up to liberate the pollen grains, what is microsporangium, how it is organized in the form of sporogenous tissue, then how this sporogenous tissue get converted into pollen grains, what is the structure of pollen grain and how they germinate. If you see the stamens, we can see here, these are male reproductive organ of the flower. They will form male gamete. If we observe closely stamens, we can see here, this is the flower of the spider plant, and this part we can see here. This is the anther part and this is the filament part, and this part is called, these are called as stamens. If we see the flower of shoe flower or hibiscus, we can see here, this part here yellow color, which are attached to a stalk with on this central axis, they are called as stamens. Here stamens, they have matured up, they have turned yellow in color. If we will touch them, and you will have yellow particles on your fingers, on your hand, and those are called as pollen grain. And here they have matured up, and here we can see again they have matured up, and they can release the pollen grain now. After they break up, pollen grains, they are released from the anthers when they break up. This is also called as dehiscence of the anther. Now, number, length, arrangement of stamens and their organization is different in different species. Like we saw in hibiscus, they are organized like this and there are many stamens. If you see here in petunia, there are 5. So they are arranged here. These are stamens. If we see here in the kachnar, they are quite big, right? This is actually ovary part, and these are stamens. So we can see here again stamens, they are big in size, and in spider plant we can see yellow colored stamens. So they are different in different plant species. Here we can see here in this flower, then here we can see stamens, they are attached to petals. We have already seen spider plant, so we can count the number also, 1 2 3 4 5 6. So in some plants they can be 6 in number, 5 in number, or they can be more than 6. So this is the larkspur flower showing stamen. So these are stamens. In sunflower, anthers, they are arranged differently. We can see here this part of the anthers is fused, this part, and filaments, they are free. This is also called as synantherous condition. Mean anthers, they are syn mean fused, and filaments, they are free. So this is called as synantherous condition. So their organization is different in different species. Number also varies. If you see the structure of stamens, right, as we have seen in the last figures, so they have a stalk, right? The stalk is called as filament, and here we can see at the top are present 2 lobes like this, right? So this part is called as anther. This is called as anther, and this is called as filament. So filament plus anther, they form stamens. They form stamens. Now when 2 lobes are there here, this condition is called as biathecous. And we will see here there are 4 groups of microsporangium, which will lead to the formation of, or we can say there are 4 pollen sacs. So we can see here, this is the filament, and which is having 2 lobes. Here also these are the stamens, and here also this part is stamens. And this filament is continued here in between, right? So if we see from the back side, so this appears like this. This is connective, sterile. So this part is sterile, and where pollen sacs they are present, this part is fertile, and this stamen, this will form the male gametes by the formation of the pollen grain. So it is called as male reproductive organ of the flower. Now we have discussed if it has two lobes, right, then it is called as bithecus, right? And if it has one lobe here, like in shoe flower, so this is called as monothecus. One lobe, monothecus, two lobes, dithecus. If we cut the section, right, here also and here also, we will see here there are four lobes of the sporogenous tissue in the dithecus anther. When four lobes are there, this is called as tetrasprangiate. Tetra mean four, sprangiate mean four sporogenous tissue. And if we see the monothecus anther, there will be two group of sporogenous tissue. So this is called as bisprangiate. Mean two groups of sporogenous tissue are present. So this is the structure of the monothecus anther. Here we can see this is attached to the filament like this. So its shape is kidney shape. And this part, which is sterile filament, this will continue and attach the lobe, whether they are two or one. So this is monothecus anther, and this is the bithecus anther. Mean when anther it has two lobes, this is dithecus or bithecus, and it will have four group of sporogenous tissue. When it has one lobe, this will have two groups of sporogenous tissue. So this is called as monothecus and bisprangiate. Now if we cut the section of the anther from here or here, right, we can see the anatomy of the anther, that is internal structure of the anther in detail. So when we cut, this is called as transverse section. When the anther is young, we call transverse section of young anther. When it is mature, this will be called as transverse section of the mature anther. Now if we see the structure here, this is anther, and this is bounded on outer side by this part. This is epidermis tissue. So I'll make it for you. So this is epidermis tissue. We can make the cells like this. Then we will make another layer. This is called as endothelium. This may have fibrous thickenings like this, which make this wall as thick. So this layer is called as endothelium. Right? This is epidermis. Now this is the endothelium. And these layers here, these are middle layers. These are middle layers. So we can middle layers, right? So this part, right, this is epidermis, this is endothelium, and these are the middle layers, right? And then if we see here, these four groups are of sporogenous tissue. So we can make the sporogenous tissue like this. We can make the diagram like this. We can complete it. This represents the sporogenous tissue. Each cell will have the nucleus. Then it is bounded on outer side by a layer. This is called as tapetum. Function of tapetum is to give nourishment to the sporogenous tissue. When they form pollen grain, total nourishment is provided from the tapetum. So this part is tapetum. This layer is tapetum, which is binding the sporogenous tissue. So we have four groups of sporogenous tissue binded by tapetum. Now this is epidermis. I'll again repeat. This is endothelium. These are middle layers. This is tapetum, and this is the sporogenous tissue. So this is again this tissue, which is made up of parenchyma or thin wall tissue. This is connective, and this is the vascular supply to the anther. I mean, this will supply water, mineral, and nutrients to the anther, which moves through the filament or connective to the anther. And these layers, which are middle layers as well as epidermis and endothelium, they give protection to the sporogenous tissue, as well as they also help in dehiscence of the anther for the liberation of the pollen grains. So we have discussed its structure in detail. We have discussed that this is the epidermis, then endothelium, then middle layer, and this is the tapetum, and this will lead to the sporogenous tissue for the formation of the spore mother cell. This will discuss here. So tapetum bounding the sporogenous tissue, 4 groups are there like we discussed. So this is all about structure of the young anther when sporogenous tissue they are intact. Tissue is intact. So function of tapetum is to provide the nourishment for the developing pollen grains. Now this tapetum can be of two types, glandular or secretory tapetum, periplasmodium or amoeboid tapetum. What does it mean? Like here we can see this is the tapetum part. If the tapetum is present here and it remains in its position, right, like here, right, and this will degenerate at certain places. I'll again make at certain places. So this can excrete the nutrients to the pollen grains, right? When it can excrete the nutrients to the pollen grains, like this will excrete the nutrient to the pollen grains or sporogenous tissue, then it is called as excretory. Glandular means this is excreting something, some secretions they are coming out, right? And when it is present at its position, like tapetum is present at its position, and this will degenerate from here, and its nucleus, they may mingle with each other, right? Here two cells, if they mingle, then this will become binucleate, and if many cells they will mingle, this will become multinucleate, right? So binucleate, when it get integrated with each other, right, during its form, you can say giving secretions, or you can say nourishment to the sporogenous tissue, right? So this is that anatomy of the mature anther. This will discuss in detail how these spores they have been produced. So we have discussed in detail that this tapetum is nourished, providing nourishment to the sporogenous tissue. So tapetum is absorptive, this is nutritive, secretory, storage and transport में भी help करता है, right? So this is glandular, or this can be amoeboid in nature. And this tapetum also helps in formation of the outer wall of the pollen grains or microspores, that is XII. We'll discuss in detail what is XII. So this sporogenous tissue, which is just a group of cells, we have discussed that this is a group of cells, right? They are closely attached with each other, which is called as sporogenous tissue. If we see the ploidy level of the sporogenous tissue, this is diploid in genetic constitution, right? So they will get separated from each other like this. They will get round off. Then these cells, they are called as microspore mother cell. Each cell will be diploid, that is 2n in number. So 4 groups are there. Now 4 groups, in 4 groups, this sporogenous tissue will get separated to form the microspore mother cell. Now each cell is diploid. These are connected with each other, then they will get separated from each other. Then each cell will undergo meiotic cell division, that is meiosis cell division, to form the microspores or the pollen grains. So when meiosis occurs, one cell give rise to 4 cells, and all the 4 cells, their ploidy level is haploid. Genetic is haploid, single N. Now we can see here, this is the microspore mother cell. When meiosis occurs, 2 nuclei they are produced, right? And then when second division of meiosis occur, 4 nuclei will be produced, right? So here we can see 2 nuclei, and here 4 nuclei, and here also 4 nuclei. One is on the back side, right? Arrangement is different. So ultimately there are 4 nuclei here also and here also, right? So when after each division, right, supposing this has divided like this, nucleus has divided and wall formation has taken place. So this is called as successive type. And when in each nucleus, we can see here, when here 2 again had divided into 2 like this, right? then we can see there is a wall formation like this. So this is called as successive. But if 4 nuclei they are produced and then wall formation occurs, this can get arranged like this, right? This is also called as simultaneous. I mean, all the wall formation or wall laying has taken place at the one place. So this is simultaneously. When after every division wall formation occur, this is called as successive. So when 4, we can say, spores, they are attached to each other, right, after meiotic cell division. So this, we use the word tetrad. Depending upon their arrangement, spore tetrads can be of different type. If 3 are visible from the front side and 1 is on the back side, then this is called as tetrahedral. When 4 they are present in one plane, this is called as isobilateral. And when their arrangement is like this, 2 are visible and 1 is here and 1 is on the back side, this is decussate, they are right angle to each other. And when their arrangement is like this, appears to be T-shape, 2 upper and 2 lower, this is T-shape. And when they are present in a linear sequence like this, they are arranged like this, this is called as linear. So from one microspore mother cell which was diploid, which is produced from the sporogenous tissue which was diploid, has given rise to 4 microspores which are haploid. So ploidy level of microspores, or which are going to form pollen grains, is haploid, and these are 4 microspores. So how we can define microspores or pollen grains? I mean, they are haploid, I mean genetic level is single N. They have a single nucleus, so this is uninucleate. They are very small in size and they are produced in large number after meiotic cell division from the microspore mother cell inside the anther or inside the microsporangia. So this development is also called as endosporous. I mean, अंदर ही सब कुछ हो रहा है. So this is called as endosporous. So this is the mature anther, from where here we have seen here there were 4 groups of sporogenous tissue, right? Now when this sporogenous tissue has get differentiated to form the spore mother cell, then spores, right? So these layers, they will get degenerate, right? So ultimately we will get large number of pollen grains here. This part will remain intact and this part is here. So we can see in the mature anther, these all the tissues have get degenerated and only 2 cavities have been left which are filled in the pollen grain, which are haploid in genetic constitution. When this anther is attached to the flower, and when the flower open up, this is exposed to the air, they start losing water because of the external dry air. When they start losing water, its wall layer cells, they shrink up, right, after losing water. When they shrink up, this will open or this will crack from the place which is weak, and that is called as stomium. So here it will break up. This is called as stomium, right? And when this breaks up, right, these microspores have already been produced, they have been matured up inside, so they will come out from this anther. Here we can see these are coming out, I mean this has split up like here, like this. So anthers, they are coming out in the form of fine yellow dust, right? So these are pollen grains, and this is the anatomy of the TS of mature anther, that is transverse section of the mature anther. So when sporogenous tissue is intact, that is young, and when it has been converted into mature, these are mature pollen grain, then it is mature. So this can days open like this here, anther can break up from here, then it is called as transverse slits. This can break up from the apical part, this is apical pore, and this can break up like this by slits which are longitudinal in nature, this is longitudinal dehiscence, right? And after release of their pollen grain, they will reach on the stigma, right? And this releasing of pollen grain and reaching on the stigma, this process is called as pollination, which we will discuss in the next lecture in detail. So this is how pollen grains have been produced and how dehiscence of the anther has occurred for the liberation of the pollen grains. Now pollen grains, they are the first cell which represent the male gametophyte, and they will form the male gametophyte. Now we will discuss what is the structure of the pollen grain or microspores, how pollination occurs, and then how they develop to form the male gametophyte. Like we discussed, microspores or pollen grains, they are released from the anther by their dehiscence, and each pollen grain is haploid in genetic constitution. They are very small in size, and they are dispersed with the help of air current or by other means, right? And these microspores or pollen grain, they are very small in size, and they are bounded by two walls. This outer wall is called as XII, and inner wall, which is thin, is called as intine, right? And this is its nucleus, which is present in the center, right? We'll discuss how this divides to form male gametophyte. So pollen grains, they represent first cell of the male gametophyte. If I make here, this is pollen grain. Now this is bounded by a thick wall, right? This thick wall is called as XII, and this thin wall is called as intine, right? All right. Now this intine, which is thin wall, this is made up of pectin plus cellulose. Chemical composition क्या है? Pectin and cellulose. So this is pectocellulosic in nature, right? And XII is made up of sporopollenin. We'll discuss in the next slide. So pollen grains, they are haploid. Their color is yellow because of the presence of flavonoids and carotenoids. They are spherical. Their diameter is 25 to 50 micrometers. Their cytoplasm is rich in starch and unsaturated oil. Starch is carbohydrate, unsaturated oil lipid, which has double bond. This is called as unsaturated oil. Then it has sporogenous tissue, which has led to the production of the microspores after meiotic cell division, like we discussed in detail, right? So this whole process of conversion of microspore mother cell after meiosis to microspores, this process is called as microsporogenesis. So these pollen grains, they are of various shapes. Like this is their outer covering, which is showing reticulation, different architectural design. So outermost layer is called as XII, right? And XII is derived from the tapetum. Like we have seen, this is the sporogenous tissue, and when they get converted to pollen grains, right, then outermost layer is derived from the tapetum. Here tapetum will form the outermost layer. So XII is produced from the tapetum, and this is chemically made up of sporopollenin. Sporopollenin is quite hard, right? So if we see its chemical composition, sporopollenin is made up of oxidative polymer of carotenoid and carotenoid esters. This is the most resistant organic material. No enzyme can degrade sporopollenin, and if there are conditions which are unfavorable, like high temperature is there, pH varies, acids are there or alkalis are there, then they are able to survive. They will not get degraded because of the outer covering, which is called as XII. So we can see here different type of pollen grain showing different architectural design on the XII. There can be reticulation, there can be ridges. So pollen grain is bounded by a hard covering, which is called as XII, but at certain places XII is absent. We can see here white point, right? So XII is absent. Where XII is not present, I mean it is not covered by XII, then these areas, they are called as germ pores. So at germ pore, sporopollenin, which is a hard material, will be absent. If we look back in the previous periods, Pollen grains of various fossil members have been preserved because of this sporopollenin and because of the exine. I mean, they can get embedded into the sediments of the rocks or sand or other sediments, and they can be preserved in the form of fossils which have become which are not present today, which were present earlier, and they have been preserved because of the presence of exine. Exine is made up of sporopollenin. Germ pores are the areas where sporopollenin is not found, and through these areas, pollen grains they will germinate by the formation of the pollen tubes, because these are the thin areas, and through these areas, cytoplasm will come out in the form of pollen tube. It will pierce through the germ pores. Now, these pollen grain, if we see, they are rich in nutrients. Some of the pollen grain, like bee pollens, people consume bee pollen as food supplement or nutritive supplement. They can be present in the market in the form of tablet or in the liquid form in the syrup. Because bee pollen, these contain all the essential amino acid, that is 22 amino acid, also rich in vitamin B12, right? And this provide energy as well as nutrients. So this is consumed by athletes for to improve their athletic performance. Otherwise, these pollen grain, when they are released from the anthers during the season when harvesting is done, right, and pollen grains are released, they cause seasonal allergies like hay fever. It is a seasonal allergy, sneezing, continuous sneezing may occur, itchiness and watery eyes, then skin rashes or skin itching. This is called a skin problem, constipation, proper motion is not coming out, bowel movement is not there, obesity lead to the formation of fat, and this also is anti-inflammatory nature. This reduces the inflammation or swelling in the body. So these are the important point or nutrient of the pollen grains. Now, when pollen grains, we have seen here, this will days open to liberate the pollen grain. So pollen grains, they are coming out. This is called as adhescence of the anther, and we have seen here the structure of the pollen grain. Here, that this outermost covering is exine. This is exine. This is exine, and this is the entine. Exine is made up of sporopollenin. This is made up of pectin plus cellulose. This is cytoplasm, having tiny vacuoles inside, and this is the nucleus, which is tube nucleus, and this is the generative cell. This we'll discuss in the next slide. And this part is germ pore, which is a thin area where sporopollenin is absent, right? Now, this pollen grain, when it is released after pollination, and this may fall on the stigma after its dispersal, and on the stigma this will germinate. I mean, like this is the female part, this is ovary, style and stigma, and this is the pollen grain which have fallen over the stigma, and then here it will germinate to form the male gametophyte, right? When how much they remain viable pollen grains after their release, this also varies from species to species. Now we'll discuss about a development of the male gametophyte. Now, male gametophyte will develop from the pollen grain, like we discussed. There are two, we can say, developments. One is pre-pollination development. I mean, when it is present inside the anther, dehiscence hasn't happened, right? In the anther, pollen grains are there. 60% of the species will liberate the pollen grain at two-celled stage. What is two-celled stage? That we'll discuss. So it is called as pre-pollination development. And when pollen grain have been released, they have reached on the stigma, then it is called as post-pollination development. So first we'll discuss about pre-pollination development. Like we discussed, structure of the microspore or the pollen grain. This is bounded on outer side by exine, and this inner wall is entine, and this part is cytoplasm, and this is its nucleus. Now this nucleus here will divide into two nuclei, right? Then wall formation will occur, and this is called as tube nucleus or vegetative nucleus, and this is called as generative cell. This is called as generative cell. So here in the pollen grain, we will see generative cell will be separated from the other cytoplasm like this. So this is called as generative cell, which is going to form the male gametophyte. So this is two cell stage, when only two nuclei have been produced. One is tube nucleus or vegetative nucleus, second is generative cell. So this is the two cell stage. So in 60% of the angiosperm, in flowering plant, pollen grains are shed at this two cell stage, that is tube or vegetative cell and generative cell. In rest of the angiosperm, they may be liberated at the three cell stage. Now when they have been dehisced open, pollen grain have reached on the stigma. If this is stigma where pollen grains have reached here, right, and further these pollen grains, they may form the pollen tube like this. So this is called as post pollination development. Now when they fall on the stigma, they will absorb water, they will get moist, and their cytoplasm will flow out. How it is happening actually? This is the pollen grain and this is its thin area. Its cytoplasm will start flowing like this in the form of pollen tube. This is pollen tube. Now when this form pollen tube, this will start getting nourishment from the styler tissue, right? And pollen tube formation is complete. Now this generative cell, right, this will divide to form the two male gametes. This will divide to form two male gametes. So a tube nucleus and two male gametes. This is called as three cell stage. This is called as three cell stage. In 60% of the angiosperm, pollen grains, they are released at two cell stage. In rest of them, they are released at the three cell stage, that is having two male gametes and tube nucleus, or when it falls on the stigma, this may form the pollen gamete and two male gametes. So this is the development of the we can say male gametophyte. This is male gametophyte, mean male gametes have been produced. So pollen grains, they form male gametophyte, and this is also development of the male gametophyte. And when they reach on the stigma, like we discussed, so pollen tube will be produced. This is carrying the two male gamete. Then this will reach toward the ovule and fertilization will take place. That we'll discuss in the next video in detail, right? In the ovules, female gametes are there, fertilization will take place. So pollen tube is glandular, secretory and absorptive. I mean this will pass here getting nourishment and reach toward the female part. Pollen tube may absorb nourishment from the styler tissue, right? So these pollen grains, which represents the male gametophyte, have been produced from the anther, that is from its sporogenous tissue, right? So we'll again revise the things what we have done. So we have discussed what is the structure of the young anther, right? Stamens, they are the male reproductive organs of the flower. Then we have discussed in detail what is microsporogenesis, that is a microspore mother cell has given rise to the spores or pollen grains, and their arrangement is different. They are haploid in genetic constitution, so their polarity level is haploid. Then it has matured open to release the pollen grain. This is open to liberate the pollen grains. So this is the mature anther. Then we have discussed after their liberation from the anther, after their dehiscence, pollen grains, they have developed into the male gametophyte. And when pollen grains are released and they reach on the stigma, this is called as pollination. So development of the male gametophyte, structure of the pollen grain we have discussed in detail. Pollen grain and their polarity level is haploid. Then they have different shapes, they are different architectural designs on the outer side. Sporopollenin, pectocellulosic in nature. Then pollen grains, they have prominent apertures, thin areas called as germ pores, and sometimes this may have germ pores may be present in the form of furrows. Then it is called as germinal furrow. Pollen grains, they can be preserved in the form of pollen fossils. Then these pollen grains, they may be used as nutritive supplements. Then they have other uses also. They may be harmful also. They may cause hay fever, they may cause skin diseases, they may cause constipation, they may cause obesity, they may be anti-inflammatory. This is a good advantage of the pollen grains. Then they will germinate to form the pollen grain. They may germinate on the stigma, or they may start germinating before pollination. So they may be released at two cell stage, or they may be released at three cell stage: pre-pollination development, post-pollination development, formation of the male gametophyte, and then they reach on the stigma and fertilization will take place. So in the next video, we'll discuss in detail a structure of the female part, that is pistil, megasporangium, and embryo sac. If you like my video, please like, share, and subscribe. And if you have any doubt, please ask me in the comment box. Thank you for watching my video.